good afternoon and good morning, our participants from US. My name is Imants Zudans, and I'm the host of this webinar. And thank you for joining us this afternoon, this morning, or whatever is your time zone. Today, we have a very interesting topic from one of our suppliers, AnalytiCon, on their unique chemical space. And I wish my computer would cooperate with me. A little bit maybe about hosts of this webinar, MolPort. We are 14 years helping scientists in R&D in companies, universities, biotechs to source compounds for their drug discovery projects. We started our company to speed up drug discovery process, and even such trivial things as sourcing samples for screening process can significantly affect how long the screening program is, and we want to reduce it, make as many compounds available as possible quickly in the right format, preferably ready already for screening at your site or at CRO site. Our ultimate goal is to get any compound scientist needs available within one business day. We have over 1,000 customers in essentially all of the continents. We list 65 suppliers in our platform. These are the most reliable and major suppliers for drug discovery process. Your orders from these suppliers, even if they are from all 65 in one order, can be delivered in one order, one package with one invoice and in the right format. As we are in 14 years in this business, we have seen what clients do, how they would like to test samples, what problems they encounter in the process of acquiring compounds. And we are full cycle service company. It starts with data. We aggregate catalogs of suppliers, standardize them, and make it available in one database. There are approximately 7.6 million stock compounds currently available on the market. We assist you in the selection process for your project. You can access data processed through Zinc platform as a Schrödinger phase database. You can access data in your 9 workflows and pipeline pilot workflows, or in software as, for example, Optibrium Stardrop. Then in sourcing, we work with projects, of course, with files that you have selected from our database compounds, but you can also use our easy-to-use online platform to look up a single compound or source a list of compounds even thousands of compounds with our very convenient list search. When it comes to ordering, we will be your partner to purchase compounds from any supplier that is involved in your order. You can make an easy order online for a few compounds, pay with credit card. There are punch-out integration options available also that are integrated into your SAP Ariba, Coupa, or other purchasing system. And when appropriate, we also participate in tenders because we understand that companies have purchasing procedures that need to be followed. For your samples, we offer reformatting. Either we take care that format is correct from suppliers. For example, AnalytiCon can prepare your samples in a range of formats, and we can help you reformat certain orders, or we also work with partners for reformatting. It can be weighing compounds, splitting compounds. You order 10 milligrams and you want 8 for storage, 2 for testing. We can dissolve compounds as well. And logistics, of course, we consolidate packages from suppliers into one shipment that is trackable with single file of all products that you can load in your software Titian or other for compound management, and the customs clearance for ease of convenience for your orders. That's shortly about us, but this is not about us. This is about great products that our partner AnalytiCon Discovery offers. And today's speaker is Dr. Lars Ole Haustad. He is a director of projects and innovation at AnalytiCon Discovery. He has worked many years at AnalytiCon, and he's a chemist by education, and got also postdoc experience working in Stanford. And with this, Lars, I give you this virtual screen. Please educate us on natural compounds and the way AnalytiCon works with them.
Thank you very much, Imants, for this very kind introduction. And I will now share my screen. Oh, I think you should have to stop, Imants. Yeah, thank you. So once again, thank you very much for this great opportunity to discuss our library capabilities, but also going beyond this in terms of what we can offer for medicinal chemistry and furthermore. So AnalytiCon Discovery, we are located in Potsdam, which is southwest of Berlin in Germany. We are a private company founded in 2000. We have our headquarters in all our resources and capabilities here in Potsdam on this very nice peninsula. So we are located right here. We have a subsidiary in Rockville, Maryland, where we do business development. We are currently 65 people and 22 of us are scientists. We offer services in the microbiology area, discovery biology, library design, and generation. And this will be the main topic of today. Also, we have a large resources in terms of separation and analytical chemistry, and going beyond this for discovery chemistry, medicinal chemistry, and structural biology and computational chemistry. So we have several programs in drug discovery. Some of these are in-house developments, some of these where we act as CROs doing the medchem. And one program I like to highlight is our services which we provide for Pharvaris. Pharvaris is a biotech company founded to provide medications in the inflammation area. And this program is now finished in clinical phase 1 and will start end of the year in phase 2. So that's a very successful program. And we have several more where we act either as CRO or where the whole program is coming from AnalytiCon. So why do we need small molecules anymore? And especially why do we need such things as natural products? So in the last 10 years, probably there were many new target classes popping up, demanding small molecules or demanding novel chemical spaces. And some of these are listed here. So in the area of small molecule RNA binders, targeted protein degradation, allosteric small molecule modulators, protein-protein interactions, and RNA epigenetics. So these are probably the hottest fields today. And these are especially the fields where we think, and many others, of course, that small molecules will have an impact beside biologicals. So AnalytiCon's collections and libraries are mainly based on two pillars. So one pillar are the libraries of pure natural products. We have a library where we isolated these secondary metabolites or natural products from microbes. And we have a library where we isolated these metabolites from plants. At the same time as we established the natural product libraries, we started also a project for synthetic libraries where we try to use the blueprints from nature, try to use the principal idea of a natural product and transform that into something which is easier to use in medicinal chemistry and easier to optimize in the end. So these are a larger library of synthetic compounds based on natural product motifs. And we have a special library of macrocyclic compounds, which also resemble natural product motifs. So this is a depiction of our chemical space. This is a principal component analysis of all of our libraries. So generated by a fingerprint, which is projected, so at 1024-bit fingerprint, which is projected into 2 dimensions, and that's what you are seeing right here. The different colors are representing our different libraries. So in yellow, you see the synthetic libraries, for example. In orange, the macrocyclic libraries. Then we have a smaller fragment collection, which is not available off the shelf. So that's an exception. And then you see in light blue, the plant-derived natural products, and in darker blue, the microbial-derived natural products. The reason why I'm showing this is that you nicely can see that these different libraries cover slightly different spaces in chemical space. So we think it's a perfect starting point for selecting the right screening compounds. how do we generate our natural product libraries? So we start with an environmental samples. From this, we isolate strains. And from these strains, we then do small-scale fermentations, which we can use in our chemodiversity profiling platform. And we are doing the profiling on an LCMS platform. From this, we are getting fingerprints, and we can look if these fingerprints resemble something which we have seen before, or if these are novel fingerprints with novel potential secondary metabolites. And if this is the case, we then do a medium scale fermentation. And from this fermentation, we then isolate the natural products, do the structural elucidation by NMR and MS, and afterwards have these isolated natural products. And we are able to do the compound management and then offer these via MolPort or others to our customers. So in terms of diversity coverage, here are depicted only the principal component analysis of the natural product libraries. So plant-derived on the left, microbial-derived in the middle, and an overlay on the right. And I just picked out or selected 4 compounds. So these different areas in this 2D depiction represent compounds with similar constitutions, so that you can really use this also for selecting interesting screening compounds. But I will show you this also in one of the next slides. So if we put this in reference, our natural products are probably the largest, or are the largest off-the-shelf available natural product library in the world. And in the middle, you see compounds which we isolated the last 20 years. So we, of course, isolated and structure elucidated more than we offer off the shelf. So these are around 20,000 compounds which we isolated in the past. And of course, the coverage is more extensively, but also an interesting aspect is that the off-the-shelf available libraries really assemble an interesting part of this space. And for further reference, you see the Natural Product Atlas, and I put a reference there also. So this is open access knowledge where you can download, I think, 120,000 natural products. And in light blue, this is the comparison to our library. And I just depicted several examples from a space where we don't cover this area. And as you can see, that's probably also the space which is not that interesting for screening purposes. So to switch to our synthetic library, there, of course, the whole design process is completely different. So here it's mainly based on the idea to take a natural product motif, to take a natural product principle. And this can be done by getting ideas from different backgrounds. So in the company, we have very diverse backgrounds of synthetic chemists. So some of us were involved in polyketide synthesis. Some of us were involved in carbohydrate, peptide, or nucleotide synthesis. All of us combines that we are natural product chemists. And of course, a huge inspiration comes from literature to get novel synthetic methods in the company, like CH activation, organocatalysis, olefin metathesis, and so on. So from all of these areas, different ideas are depicted and sketched, and then defined as a library design. And of course, we are following the usual guidelines like Lipinski and Weber rules. But we also can use the feedback we get from a very diverse customers over the last 20 years. And this extremely helps to redefine our objectives and to stay really on top of the game. So, of course, as I depicted on one of the first slides, it's very important to address new target areas, and that's a dynamic process where we always try to incorporate new scaffolds which can address these new modalities, like the RNA small molecule binders, for example. And if this design idea is there, then we do a feasibility study. So we try or probe for the synthetic route. We probe for the chemistry scope. And then we do a feasibility on the compatibility of the functionalities. We develop a purification method. We look for stereochemical integrity and stability. And if all of these gets a green flag, then we go for production. So we synthesize the starting materials, the library intermediates, and of course, finally, the final compounds, do the purification by HPLC, and then the QC afterwards. So during this journey for the last 20 years, we covered a very broad range of different natural product scaffolds where we populated these synthetic libraries in. So some of our libraries are peptide-based, some of our libraries are alkaloid-based, some of these are macrocyclic libraries. Then we have a large area of scaffolds where we don't have a clear classification. We have terpene-based and carbohydrate-based scaffolds in the library. So this is not a painting by Jackson Pollock. This is just color-coded. So for the different libraries, so in total we have 123 scaffolds in this library of 32,000 compounds. And on this slide, you see some of the examples in this principal component analysis and of course in the library. So as you see, it's a very diverse set of scaffolds. And also what you can see here, the distance in this projection also resembles similarity in scaffolds. So once again, that's very helpful for selecting compounds. To get an impression on the three-dimensionality of our compounds, we did a PMI analysis, principal moments of inertia. And what you get from this triangle diagram is, of course, the distribution between shapes of the compounds. So first you have to do a force field calculation, and then you can do this depiction. And you see which compounds are lying or are nearer to a rod-like shape, which compounds are nearer to a disc-like space, and which compounds are near to a sphere-like space. And as you can see, that's a quite nice distribution around these geometries or these shapes. Just for comparison, the RD fingerprint is referenced on the right-hand side. So for the macrocyclic libraries, we try to incorporate as many different and diverse scaffolds as possible. And for this, we developed quite efficient macrocyclization protocols, which include ring closing metathesis, a very unique and proprietary two-component cyclization strategy, where we used epichlorohydrin and an amino sulfonamide to build up the macrocyclic scaffold. And we used classical macrolactamization chemistry to cyclize scaffolds in a very modular way. On top of this, we developed a technology which allows to even further increase the scaffold diversity of the macrocyclic library. This has been done by defining 3 building block pools, which we combine by solid phase chemistry to an acyclic precursor. And this acyclic precursor is afterwards then cleaved from the resin and put in 96 well plates. And in this 96 well plates, we are looking for the cyclization reactions. So we're doing the cyclization reactions in 96 well plates and can directly by LCMS see if these acyclic precursors cyclized. And if they did so in the right way, so not forming a dimer, then we select these scaffolds for scale up and do gram scale synthesis of the macrocyclic scaffolds, and afterwards do decoration chemistry to synthesize the final compounds in the end. So that's a very modular, medchem friendly and scalable chemistry. And together it allowed to put together a library of 4,600 compounds, macrocycles, coming from 152 scaffolds. So that's I think a very great achievement. That's a very scaffold diverse library. And once again, there are a few examples from this library depicted in this 2D diagram. And once again, the scaffolds are color coded to differentiate the different scaffolds. So on this slide, you just see a reference to natural occurring macrocycles. And in this 2D diagram, they are depicted in light blue. So all of these have been isolated here at AnalytiCon. So together, plant-derived and microbial-derived, we isolated over the years around 940 different macrocycles. And 2 of these are depicted in the diagram. And as you can see on the right example, there's probably also a reason why we didn't include these in the synthetic libraries, but because they are very complex and hard to optimize. But if you look at the left-hand side, that's a scaffold looking much more similar to the scaffolds we provide from our synthetic program. So we think we have a very nice coverage of the more useful macrocyclic scaffolds having somehow natural product background. And once again, just to see also, or to get an impression about the shape of this library, we did a principal moments of inertia analysis. And on the left-hand side, you see the synthetic macrocycles, which are available off the shelf. And on the right-hand side, you see the natural macrocycles, and only a part of these are available off the shelf. So what can you do with all these similarity and diversity analysis? So of course, you can really describe chemical spaces which are wanted and which are also unwanted. You can do similarity and diversity selections. You can really describe chemical spaces in which you're interested or which you want to avoid. you can use this fingerprint-based diversity also to opt out chemical spaces which you are not interested in. And you can, of course, go beyond this. So we can do also can do pharmacophore-based selections, either ligand-based or based on crystal structures or structural biology information. And furthermore, we can do multiple parameter optimizations, and an example is shown on the next slide. So we can use wanted set of parameters to define a space which we are interested in. And this example is coming from Pfizer, from the Wegner et al. Paper from 2016, where they used several descriptors to define this multi-parameter optimization. And using this algorithm, we selected from our off-the-shelf available libraries a set of natural products, a set of macrocycles, and a set of synthetic compounds, which potentially are interesting in the CNS area. And these, of course, are available off the shelf, and you can cherry pick from these selection as you like. So last but not least, I want to show you one application of our chemical space. So fragments from nature, as I said, these are not available off the shelf. These are only available in projects. And once again, the principal moments of inertia analysis of these fragments and principal component analysis. And as you can see, it's nicely distributed in terms of shape, but also in terms of diversity. These are around 2,000 fragments which are available in projects. And the unique point that we address with our fragments is that we have this natural product background also in our fragments. So we use also a natural product motif even in these small compounds. So they are a bit larger than conventional fragments, but what you gain from these is much more information. And these are all examples where we got a crystal structure from our fragments in two different projects, endothiopepsin and protein kinase A. And as you can see from the depiction of this pharmacophore, we get a lot of pharmacophore information just from a single fragment. And that's kind of unusual that you can directly define a pharmacophore, a meaningful pharmacophore from one fragment. And this is really the case from our fragments in a range of different projects. So one especially interesting example are these two fragments of the project of endothiopepsin. So this is an aspartic protease. It's a model system for pepsin-like proteases like base and renin or renin. We just selected two fragments from the many crystal structures we got in this project. And on the left-hand side, you see a fragment which is covering a complete different part of the binding site as the example on the right side. What is interesting is that the overlay is nearly perfect. So, and we have a amine and an alcohol functionality, and they inherit more or less the same space. So the idea was really to combine these. And we did this by combining these two fragments in one compound, which we call the merged compound. And we were very lucky to get a crystal structure also from this merged compound with a very high resolution. And what could be shown is that the by ITC measurements that the KD of the fragments, which in both cases were single-digit millimolar, could be increased to 2.7 micromolar. So we gained a magnitude order of binding affinity. And what we also could show is that the merged compound assembles or resembles nearly the same binding mode than the two fragments, which is very unusual, and it's a very unique example of a merging strategy which really worked out in the end. So as a summary, AnalytiCon's unique chemical space is available off the shelf. So as I said, we have the macrocyclic compounds, which is 4,600 compounds, the synthetic compounds of 32,000 compounds from 123 scaffolds. The macrocyclic scaffolds are 152. And we have the plant-derived natural products and the microbial-derived natural products. So with this, I'd like to end. And of course, I'm very happy to answer any questions.
Thank you, Lars, for a very insightful presentation. Of course, this is just a quick glimpse in your expertise, in your capabilities, and it's not possible in such short time to explain everything. Mm-hmm. So for maybe listeners, if you have, if this is something interesting to you, get in touch. We'll connect you to Lars to explore deeper chemistry questions specific maybe to you. So to maybe switch to questions, we do have a couple of questions. First, maybe about compounds themselves. So you mentioned that compounds are available for individual purchase. They can be cherry-picked essentially from sets, but also you have sets available as sets if you as well, right?
Yeah, so sets we seldomly put on sale. So it's really more cherry picking from the whole library, and that can be done via my MolPort or it can be done directly. So and we are both, I think, able to really help you out with the chemoinformatic services you might need.
What is the quantity of compounds available? Are these available only in milligram scales, or maybe a few are available in hundreds of milligrams for follow-up experiments? Could you comment on that?
Yeah, definitely. So for the whole library, typical amounts for screening which are purchased are in the area of 0.5 milligram to, let's say, 20 milligram. 20 milligrams, very rare. But this is really meant as a screening offer. And for the synthetic compounds, all the protocols are enabled. We develop these libraries so we can easily scale. And in many projects, we scale single compounds up to 100 gram. And if more is needed, then we have partners in place to really do then route development and do scale up procedures.
And could you comment maybe a bit more of how you calculate principal moments of inertia?
Yeah.
Could you comment a bit on those data?
Yeah, glad you asked. So all of this has been done with NIME using several vendors, mostly the open access available tools from especially RDKit and so Novartis and Vernalis. What is behind this is that you first do a force field calculation. In this case, it's a very simple force field. And you get the lowest energy conformation, or you select the lowest energy conformation. And then you look for the medium in terms of the shape of the molecule and the deviation from this medium or a median. These are then in principle 3 principal moments of inertia. And that's why it's a triangular diagram in the end. And you can directly see these rod-like similarity or sphere-like or disc-like. So I think it's a very nice depiction of shape diversity. And what is also quite interesting is to probably look further down the road also in terms of dynamics. So do they behave different in different environments? So if you have available force field calculations in different solvents, you can even do that and see if the shape distribution switches from one depiction to another.
That was a very interesting plot, because there is a lot of discussion about that commercial compounds tend to be flat, and how to increase their essentially 3D properties and sp3 hybridization, etc. So it's a very convenient way to show that. I know that KNIME community tends to share workflows. Is there any chance that this workflow is maybe available from you or on KNIME to calculate?
Yeah, absolutely. So because I only used open access nodes for this PMI workflow. So we also licensed nodes from other software vendors, but in this case, I think it's not necessary. So you can just use what is there, and I can definitely provide these. Yeah.
So if anyone is interested, get in touch with Lars. For the compounds, for about getting to chemistry, let's say you have certain scaffold. How does it work with AnalytiCon? Can someone take your scaffold and come to you for modifications of this scaffold?
Yeah. So first of all, these are non-exclusive, off-the-shelf available compounds. So there are no strings attached. But of course, we developed these scaffolds, and it's, I think, a good idea to ask if we have some ideas in the direction you like to see. So especially because we did typically not one library from one starting material, but several. So there are a lot of different chemistries available, different exit vectors, and we also know what direction works, which reaction works on these scaffolds, and which ones don't work. So we are absolutely happy to provide you with follow-up services in terms of discovery chemistry, or going even to medicinal chemistry in the end.
All right. So you have very nice collection of very interesting 3D shaped compounds, off-the-shelf compounds available for cherry picking in multiple formats. You can also work on cheminformatics projects, on follow-up projects. And yes, I think we've covered essentially all of the main questions. For further questions, you can contact us at sales@molport.com. We'll get you in touch with Lars. This webinar will also be available in a recorded version for you to maybe go back to look at some portions of slides, share with colleagues, or also to people that registered but for some reason couldn't participate. And as a last point on the agenda is there will be a brief survey after the webinar that will drop in your email. Please fill it out. It will help us to understand if this was useful, if we should continue such webinars to explain our approach, to explain how our wonderful suppliers work, because we really, MolPort is just a platform. Lars here is the chemist, and AnalytiCon is the company that provides these wonderful products. And so please fill out the survey to help us understand in which direction to take this. And with this, I would like to thank Lars for your presentation. Thank you everyone for participating. Have a good evening or have a nice productive workday, and stay safe. Bye.
Thank you very much, Imans, for this very nice webinar. And also Christine. Thanks a lot.
Bye.
Bye.